my annotations. So, in the micro lab, they also, I talked about how they also have PCR and things like that. Why do we have PCR in the micro lab when something can easily get grown on a plate? Because not everything grows easily, right? When you learned all of your micro stuff and watched all those sketches, those were literally a lifesaver. I'm not gonna lie to you. I'm super visual, need like little cheat sheets, like make me remember stuff. Best thing ever. Anyways, not everything grows. Yeah, so more accurate and precise than biochemical testing. And if you know anything about anything and you went through your rotation and you're learning your rotation and in student lab, bacteria don't read like a book. Like, even though it's supposed to be such and such positive, well, maybe this one isn't. Sweet, right? It's a great time. So PCR actually helps us identify. Molecular testing helps us identify organisms a little bit better than all those biochemical reactions with catalase and all that good stuff and fun things that I don't have to do anymore. It's a great time. Being in the molecular lab, I don't have to deal with the smelly plates and the gross things that grow on them. But it is fun to go in there, I will say. They get weird stuff sometimes, and like I love to go in there and look at the parasites that they get. It's a great time. I will say that our lab is boring compared to micro, because they get some random stuff. So it analyzes the genome, transcriptome, and proteome instead of microscopic colony morphology, pigment production, fermentation. They're still used though, because it's cheaper to grow something on a plate than it is to do PCR. PCR is super expensive. And targets can include bacterial DNA, viral DNA, and RNA, and prion proteins. That is also great because prions don't grow on plates. What are prions? Infectious proteins, right? So this is the most common, like commonly known one that causes what? Mad cow disease, right? It's a great time. You can't grow those, so it's only PCR methods. Now we don't do any of that testing. No, thank you. So target microorganisms for molecular-based testing. Again, if you're going to develop a molecular-based test, you want ones that are difficult or time-consuming to isolate, like mycobacteria. Hazardous organisms. You don't necessarily want to be growing something that is hazardous so that you can contaminate everybody else with it, right? That's probably, let's stay away from culture on that one. And those without other reliable testing methods, like HIV and HCV. High volume tests also are great, right? If you're constantly getting tests for gonorrhea or tachymonas, there's so many tests. Chlamydia, we don't get them here because we're not like a women's health hospital, but like regular hospitals, like that have like women's health clinics and things like that, like they get that kind of testing regularly. Like, yes, high volume. We don't do that here. But if they do, it's nice because you can batch them all together, run them all at once. You don't have 96 plates growing. You don't have like all these IDs to do. You just slap it in there, you get a result. Especially if you know what you're looking for, right? Like as far as like STD testing and things like that, like if you're looking for a very particular infection, it's easier to just try to find and do PCR on that one particular thing than try to grow, and who knows what the heck you'll grow. Could take days to grow, and then you gotta like do all those biochemical tests on it and everything. Like, yeah. So applications of molecular-based testing, again, rapid and high throughput identification of microorganisms. They may not require culturing, isolation, or biochemical testing. Again, yeah, we don't need to do that. We just need the body fluid that you're getting. Like, we can detect small quantities. We don't need to grow it up first to detect it. We used to do viral culture here. and shell vials. We don't do that anymore. It's all PCR stuff now. They got rid of a lot of it during COVID because they didn't want to grow COVID and spread it. So they got rid of a lot of viral shell culture and virology during COVID, and it's a lot. It's molecular-based methods now. So you can also detect and analyze specific genes for resistance, like MEK-A, VAN-A, beta-lactamases, right? If you want to know if an organism has a gene for those antibiotic resistance present, you can run PCR on it. Genotyping. Again, we can do 16S genotyping. So it's ribosomal RNA, and we currently send that out to the University of Washington for things that don't grow, right? Like, so if we get a body fluid and nothing grows in it, we'll send that same body fluid out for 16S sequencing to see if they can identify whatever it is. And again, discovery of new microorganisms. Specimen collection, you got to preserve it. You need to have viable nucleic acid, and it has, hello?
You actually,
Yeah.
there's treats that I made.
You made treats.
And I also made caramel apples on berries. I don't know. They're in my office. These are eyeballs, truffles. These are Oreo truffles. And then these are Nutter Butter Money cookies.
Oh, I want something to make me feel better about the apples. Mary brought in cupcakes from my things that they dropped them on the lid. So yeah.
I worked hard on the apples. I don't know why the caramel just kept coming off.
We'll eat them and destroy the evidence. Don't worry.
So I brought more caramel that you guys could dip them in, but I'll bring them back later.
Okay. Listen, we can snack all day, bro.
Yeah, dig in.
Yeah.
Pass them around, whatever you want to do.
Okay. I know, right? I will say that we do get improperly collected specimens all the time. If you, oh, you're taking a picture. That's cute. You haven't done your micro rotations yet, but a very common swab in micro is an e-swab. Just for use for bacterial culture. We need a flock swab in viral transport media. They like to exchange them both. The viral transport media tends to kill bacteria and stabilize the viruses. And the e-swab is stable for bacteria, but not necessarily for viruses. Make sure that you are following the proper collection procedures. They're like, it's a swab. Why does it matter? It matters. Okay? It's blood. Why does it matter what tube it's in? It matters, right? Just like it does for chemistry. It matters. It can make a difference on your analysis. Your analysis, not your analysis, but like depending on the collection and the ions that are in there. There are commercial collection kits available. During COVID, lots of places went with commercial collection kits. We did not. We made all of our own collection kits and had volunteers put together collection kits for COVID, and it was a great time. The Clinical and Lab Standards Institute has guidelines for proper specimen handling. Please follow them. And CAP requires procedures for specimen handling, collection, and transport. Wait, other people made things too?
Oh, do you guys want to? Do you want me to? Yeah.
Oh my God, I'm so excited right now. What do you mean?
This is better than school parties. Yeah, I know.
Way to skip her. You can't leave people out, bro.
Yeah.
I'm excited for you to come to me. I'm not gonna nap. This is all getting recorded, isn't this sweet?
Yeah.
I know.
I'm trying to keep my.
Oh my God.
But,
This is so cute. Sorry, next.
I don't know if that's like a.
I know, I'm gonna give it to him. There you go. Merry Christmas. Happy Halloween.
I'm trying.
Okay. So we can use sterile containers for collection, swabs, specialty systems, proprietary systems, anaerobic transport systems, viral transport systems. We use a lot of viral transport. We don't collect a whole lot of bacteria. But yeah, make sure that you're following the collection guides if you are out there collecting. I know some labs that you may get a job at require the lab techs to go out and collect on the floor. I know that, especially if you are working an off shift. So I know Akron Children's does. If you work second or third shift in CCL, you're required to go do draws on the floors. So make sure that you are following proper collection guidelines. That is still part of your job, potentially, wherever you work. Did you guys phlebotomize each other yet?
Yeah.
That's so fun.
Yeah.
Sample prep. Consider the specimen type. More rigorous lysis. We talked about that. It's required for organisms with cell walls, or if they're in FFPE, or if they're in stool with lots of inhibitors. Consider the number of organisms in the sample and concentrate by centrifugation if necessary. So for respiratory specimens, like a BAL, sometimes we spin them for if we are running M-pneumo or CMV because they tend to be intracellular. And we want to concentrate the cells. We don't necessarily want all of the extra wash fluid that comes with the bronchoalveolar lavage. So we centrifuge it, get a cell pellet, decant the extra, and then resuspend in a smaller volume so that we can concentrate everything. Inactivate or remove inhibitors. Again, definitely an issue in stool. There's lots of inhibitors in stool. There's also lots of inhibitors in sputum. And then in CSF, we do get some CSF samples, but the regular process that we have doesn't have an issue because of the way that the extraction is for our CSF samples. Hemoglobin and products of metabolized hemoglobin can inhibit DNA polymerase. And then inactive RNases. We need to do that. So we talked about selecting primers. Here's just a pictorial representation of good areas to pick. Here's our target organism right at the top. We have genomes A, B, and C, right? Those are the genes on the genome. If we have target organism, and this is a variant of it, right? It has A and C, but it doesn't necessarily have B. And then other flora, they share A. They all share A together. We would not want to pick A if we were trying to target that organism. If we wanted all variants of this organism, which one should we pick? C, right? Because B is missing. If we only wanted this specific subtype of this target organism, which one could we pick? B, correct. So again, blasting your stuff and picking probes and primers is important. So PCR quality control. PCR and other amplification methods are extremely sensitive and very specific. For accurate test interpretation, we need to run controls. So we always run a positive control, right? It goes through the entire testing process. It goes through extraction, goes through amplification, goes through detection. It has to go through the same process start to finish, because if something happens, you need to know where an issue happens so that you can rectify it. You don't want to release patient results if there's something wrong. Okay, we also run a negative control, right, with everything to ensure. So we have a negative control that goes through extraction and amplification, but we also run a second negative control. It does not go through extraction. It only goes through amplification. So the three things. If your positive control shows an assay system is functioning properly, usually a lower and upper limit of detection. If it's a quantitative assay, you need to show and demonstrate that the whole linear range is working. So you need a low control and a high control. We call them low AMRs and high AMRs. Low analytical measuring ranges and high analytical measuring ranges controls. The negative control, again, detects the presence of unwanted targets, but should not react to desired targets. So because this negative control goes through the entire process, including extraction, it will have an internal control added to it, right? So we know that the extraction process worked. There is a control for that extraction process, and that also controls for the PCR process. It will be negative for the viral target if you don't contaminate anything. Okay. An amplification control, we call it no template control. So you're not adding template DNA to this control. You're adding the master mix, and you're adding water, DNA RNA-free water. So the amplification control ensures lack of a product in an amplification procedure, and it's truly due to the absence of the target organism and not the presence of inhibitors. So, like, hold on. I'm sorry. This is your MTC, the reagent one. We call that the reagent one. I don't like this. We call these things different. This is your NTC, your reagent blank. Monitors for contamination, contains all of the assay reagent except for the target sequence and should always be negative. The amplification control, this is all amplification. These are all amplification controls. Okay. You have positive amplification control and a negative one. Okay, so you always have, this is your internal control. That's what we call this one. In the lab. Your internal control says it's in every sample, right? You add it to ensure that there's not an inhibitor present. It should be there if your sample is truly negative, right, for the viral target. It doesn't have to show up if your sample is positive for the viral target, because again, why?
In a low concentration.
And what happens if you're positive for the viral target? It uses all the DNTPs and things, right? It uses the polymerase, it uses the DNTPs. There's not enough left over for this internal control to amplify. But we know if it's positive for the viral target, amplification worked. And we know that if it's positive for the internal control, the amplification worked, right? So then you have this negative, no template control, right? That's where we add the master mix and the blank, the water, because we want to make sure that we have no contamination in any of our reagents and that it's good to go. If something amplifies in this, we'd be in trouble. Okay, we got troubleshoot, we got decontaminate, we got to do some stuff. Okay. Also, if the viral target amplifies in the negative control, that's also a problem, and we need to decontaminate. So yeah. Okay, so PCR internal controls. It monitors proper isolation, amplification, and detection. It goes through that whole process of extraction, right? It goes through the whole process of PCR. You can have lots of different internal controls. You can have homologous extrinsic, which means it's target-derived control with non-target derived sequence inserts, and it controls for amplification. So if you look at the picture on the right, here's your target organism, okay? We have a plasmid. It's saying we're going to take this region of the plasmid. So it's target derived, it's part of the target virus, like it's in the it's a plasmid in there, right? But it's when a non-target derived sequence is inserted into it. So we're not looking at this sequence, but it's in there. You're inserting a plasmid with a known region, okay? A heterologous extrinsic is a non-target derived control added before extraction. controls for extraction and amplification, but requires its own set of primers. We do this. For all of our infectious testing, that's what we do. So, and a heterologous intrinsic is non-target sequence naturally present in the sample, such as a human gene control. That's for our like Behens assay. Remember, we had that actin beta. We wanted to ensure that's non-target specific, right? We're looking at beta, but we know that the extraction worked if we got human DNA also. Does that make sense? Yeah, so this is like the Behens assay with the actin beta. Does that make sense? Questions? No? Okay. So quality control false positives. Possible carryover, contamination. Check your reagent blank. Okay. If your reagent blank is still negative, that can happen. You can contaminate from sample to sample, and your reagent blank can still be negative. Let's say you're pipetting template DNA into one tube and you have two tubes open by accident, right in the hood at the same time. And there's a little bit of aerosolization. It gets into the second tube, right? It might not have gotten into your no template control blank, but it might have crossed into your patient next to you. So we look and monitor this in a variety of different ways. We really are diligent reviewing patients that are next to very high level viral load patients. So if we have a sample that has like 10 billion copies right here, right? And then this next one is negative, typically, right? We run our HEMOC kits for viral loads twice a week, right? We kind of know you're not going to go from nothing to 10 billion overnight, right? You shouldn't. Ooh, you'd be in trouble. But like, you would see a slow increase over time of their viral load if they would. But let's say this one was 10 billion and all of a sudden this one was negative the day before and now we're at 2 billion. Well, that doesn't seem right. So like we look at previous results, okay? If we're calling a new positive, we look at previous results. We look at what's next to it on the PCR run, right? And we say, okay, well, this one was pipetted first. Is there a way that this one could have contaminated that one? Potentially. So what do? We start over. We do PCR all over again with a fresh sample. Okay? And we check its validity before we release a result like that for somebody who was previously negative. Now, if we pipetted this one first and this one was 10 billion and this one was 40 billion, this one likely did not contaminate this one. We pipette in one direction. We always start left to right. And then this one should be closed and all that. So typically though, if you have bad pipetting, you're gonna end up contaminating your negative control. If it's you who sucks at life, you're gonna contaminate everything. It happens. There are people who suck at life. Mistakes happen. It's okay. But then you don't release any patient results on the run and you're like, oh, can't do that. I gotta rerun them all. Dead and dying organisms, retest 3 to 6 weeks after antimicrobial therapy. Again, you're not going to get active DNA like that's ready to be analyzed if you're on antimicrobial therapy, right? Because you got antibiotics in your system. The detection of less than clinically significant levels. We talked about that. We can detect down to 5 viral copies in some assays, but some aren't as sensitive. Sometimes we can only, our lower limit of detection is 500. You know, it depends on how sensitive the assay is, the specimen type that we're isolating from. So like, if you have less than 500 copies, it's probably not relevant anyways. So, quality control, false negatives. Organisms may be present, but nucleic acid was degraded during collection, transport, or extraction. We talked about that. If your internal control doesn't show up and your viral target doesn't show up in PCR, something happened. We re-extract because it could have been us. Maybe while we were bleaching our cap doohickey, our cap opener. We got bleach in the specimen. Well, that sucks, right? But it's gonna inhibit PCR, I can tell you that, right? So we gotta start over. We try to re-extract. It could potentially be a collection problem. Okay, what if they didn't let the alcohol dry long enough and we only got 3 drops of blood? Which happens, you know, it's gonna inhibit everything. Sometimes they're on drugs as well, and that does some funny things. Like if they're getting some random concoction of drugs, that can sometimes inhibit things. That's what the medical directors are for, to review the chart and see what they got going on to see if it would inhibit the PCR. But yeah, so amplification inhibitors, certain anticoagulants can also inhibit PCR, those anions and cations, and then hemoglobin can. Viruses, classic methods of detection include antibody detection, antigen detection, and culture. We don't do culture anymore. We don't want to grow viruses to spread them everywhere. It's a great time. We used to even we were digging through a -70 here like a while ago, liquid nitrogen freezer or whatever, even colder than -70. Like polio and stuff and cultures, everything here. Crazy. It's a great time. Disadvantages. Immunodeficient patients may not produce antibodies, right? If you're doing like classic methods of antibody detection, antigen detection, antibody testing requires samples during acute and convalescent stages. Did you learn about that? Acute versus convalescent in chemistry? Did you do you did? So that you can see an increase in titers of your antibody and levels of your antibody in your system? I don't remember. Maybe that's hematology also.
We still have immunology. So.
It'll definitely be in there. It'll be in that. So if you are actively sick versus recovering versus not sick at all, right? Your antibody levels are going to change over time. So making collection, if you're doing antibody testing at a time where your antibody levels are relatively low in general because you're not having an active infection, that might not be the best time. Timing is everything with those kind of things. And then culturing just takes time. PCR, day, done. Molecular methods include target probe and signal amplification. Tests are designed for identification of viruses, determination of viral load, and genotyping. So when we do viral load, it's used for disease prognosis and efficiency of antiretroviral therapy. So a lot of these HEMOC kids, because they have terrible immune systems, they're given antiretrovirals, right? They're not given antibiotics because they're viruses. Different method. Yes, we know the difference between viruses and bacteria. Yes, what's the difference? Okay. Define alive. And replicate. Well, they can sit on a surface and infect you for a long period of time, depending on the virus, because they're encapsulated and stable. So they don't need to be in a host to necessarily be alive, but to replicate and propagate itself and infect other things, yes, you need a host. Whereas bacteria, they're an all-in-one. They got the whole shebang. Okay. So viral load tells you the number of viruses per mil of body fluid, and it's measured with qPCR, digital PCR, other methods. Again, real time is what we've used, and measurements by different methods may not be equivalent. So technical variability and biological variability. So I will say that we have viral load monitoring that we monitor in international units per mil. And we have viral loads that we monitor in copies per mil. The difference, things that have international units have international standards that have been developed. Because there's so many different subtypes of viruses and things like that, right? They all amplify and replicate at different rates. People's PCR reactions replicate at different rates, and they are more efficient than others, right? So there is something called an international unit. So they have international standards that are derived for everybody to use all over the world and compare it to, so that everybody's result, no matter where you go, should be somewhat similar. Yes, there's still going to be variability, but especially based on maybe subspecies of the virus or whatever, there's still going to be variability, but they should all be close. Now, there are viruses who don't have that because they are very difficult to characterize. For example, our adenovirus assay. There's so many different serotypes of adenovirus. They don't have an international standard for it. So we set up our own using own standard curve, using standards and everything. And we still have to fall within an acceptable range on our CAP surveys, right? But the range is typically a little bit more giving than the ones that come with international units per mil. So like, let's say the range of acceptability on these guys is like plus or minus 2 logs, which is huge. This one might be plus or minus 4 logs. It's really not that wide. But what's a log? 10-fold dilution, right? So let's say like plus or minus 4 logs is a huge difference, right? You can go, if you have 4,000, 4 to 40,000. 4 is 4, right? 1, 2, 3, 4. Plus or minus, that's minus. That's 4.0. Plus would be, right? That's a huge range. They're not going to be that wide. But what I'm saying is there is an acceptable range that's a little bit wider and more forgiving for things that don't have an international unit as a standard, because there's so many different serotypes and so many different things that are very technically different based on your method or your lab developed assay, what serotypes you're using, what standards, and biological variability within the virus itself. Test performance features of viral load. So sensitivity, the lowest level detected at least 95% of the time. You run 20 samples. If one does not meet the criteria, that's your 95%, right? Acceptable limit. That's your limit of detection. If you say my limit of detection is going to be 500 copies, you better detect 500 copies in 19 out of 20 samples that say that they have 500 copies, right? Accuracy, the ability to determine the true value, how close you are to the true value. If the viral load is supposed to be 3,000, are you 3,000? Or are you not 3,000? Precision, reproducibility. If you got 3,000 the first time, did you get 3,000 the second time? Running the same assay on the same method, using your same reagents, right? Specificity, negative samples should always be negative. Positive samples should always be positive. I will say that a lot of times when you take a culture method or something else and you transfer it to a PCR method, PCR is more sensitive, right? So you may pick up more positives that were initially marked as negative when you switch to a more sensitive method, which is completely understandable. You would have somebody else, an external lab, run the same test to see if they got the same result, right? That does happen. Linearity, a serial dilution of a standard curve closely approximates a straight line. You should only be using the linear part of the curve to make your standard curve. And flexibility, accuracy of measurements of virus regardless of sequence variations. So even if you have a little bit of variation, does your probes and primers, do they still pick it up? So antimicrobial agents, you have static, which inhibits growth, like anything like bacteria, static. Okay, bactericidal kills organisms. Classified by static or cidal, they are the mode of action, right? Do they break down the cell wall? Do they do other things? Like, do they, I don't know. I can't think right now. What are other mechanisms of like cell death? Since they break down some poke holes in the cell wall, some like just, it could affect transcription and translation. Oh, some make it explode, like they get hypotonic or hypertonic, hypo. Those are words, right?
Yeah.
It's a long day. Chemical structure, you can alter the chemical structure of them. It should be selective for the target organism with minimal effect on mammalian cells. So if you're going to use something that's antimicrobial, to get rid of bacteria and viruses, you better hope it doesn't kill you also, right? Like, that would suck. Although bleach is kind of corrosive, so, protect yourself. But, distribute well in the host and have a long half-life. So if we're talking about internal drugs like antibiotics at this point, you want it to be distributed well in the host and again have a long half-life. Why?
Time to do its job without like going away.
Yeah, it needs time to like find the bacteria, kill the bacteria, right? Like the virus, whatever it is. And it needs to get to where the infection is. Right? Sometimes it's not just chilling in your stomach. Sometimes it is. What's the bacteria? Yes. Right? It's right there. You just easy. You just take a pill. I don't know. I've never had it. So I don't know. But I would assume that that's an easy one to treat because it's like right in the stomach. Although there is stomach acid, so that might deactivate some antibiotics. I'm not sure. Never had it, never had to take medicine for it. And Cidal has activity against a broad spectrum of microorganisms. Some are very specific, right? Antibiotics can be very specific sometimes. Others just like, it's like a blanket and you're just killing everything. You can attack protein synthesis. You can attack metabolism. You can attack cell wall integrity, membrane integrity. Again, different sites of action. Things that disrupt the cell wall. Beta-lactams, penicillins. I'm not gonna lie, this is the hardest part of my life in micro. I cannot remember the drugs and anything that they did for the life of me. I will own that. That is my struggle point. Okay? I'm not on antibiotics regularly. It's not my jam. Like, but I'm sure you guys are much smarter than me. But you got beta-lactams like penicillin and cephalosporin, and then glycopeptides like vancomycin. Disrupt cell membrane structure or function. Polymyxins. Inhibits protein synthesis. Aminoglycosides. Like gentamicin. Tetracycline macrolides and lincosamides. I don't know. Clindamycin. It's clindamycin. That's all I know. Inhibits nucleic acid synthesis or integrity. Quinolones. And then inhibits metabolic synthesis. Really, again, I do the testing, I don't do the treatment. In my opinion, although it's wrong, it's a doctor to get the degree in what to treat somebody with, not a medical lab scientist, but it's on your exam. So you need to know it. It will be on your MLS test as well. So study it and don't forget it, and then you can just. Okay. Mechanism for the development. I guess it kind of matters though if you're looking for like antibiotic resistant genes. Like you need to know what ones are common if you're the one doing testing in different microorganisms. So I guess I shouldn't sit down. You need it. Keep it. It's a great thing. Mechanisms for development and resistance to antimicrobial agents. So we have enzymatic inactivation. So again, beta-lactamases, altered targets, mutations in the gene encoding for penicillin binding protein. So if they mutate to not be affected by penicillin, well, sucks for us. Can't use penicillin anymore, right? They're evolving in response to the things that we're producing. That's why we have this huge antimicrobial resistance superbug problem. We have our own superbug here. She knew we were going to talk about this, so she wanted to be an example. We have altered transport of agents in or out of the cell, and some bacteria can actually change their outer membrane proteins, like their porins, to decrease influx of antimicrobial reagents. So they'll like spit it right back out. They won't let it in. When they acquire these genes, they can pick them up from other resistant organisms as they are in the environment. So viral genotyping, viral genes mutate to overcome antiviral agents. We can detect that by sequencing. Primary resistance mutations affect drug sensitivity, but may slow viral growth. So they can still be effective to some sort, but they're not going to be 100% effective. And then secondary resistant mutations compensate for the primary resistance growth defects. So change drug therapy for patient once a primary resistant genotype is identified to avoid secondary mutations. So sometimes they'll start somebody on an antibiotic just to try and get like some relief of whatever infection they're fighting. And then they'll find out what exactly it is, if there's any resistance to it, and then modify the antibiotic regimen after that. Does that make sense? Okay. Advantages. So clear evidence of resistance. MIC is near the breakpoint of resistance. So did you guys talk about breakpoints and stuff? Yes. Okay. Can directly test the clinical specimen and then monitor the spread of resistance gene in multiple isolates for epidemiological investigations. So we're using molecular methods, right? We can compare lots of different isolates quickly for resistance and gene modifications. So we don't have to wait for it to grow. What can happen when something's growing potentially? It's replicating, so what's happening to its genome? It could be acquiring mutations, right? So if we get the direct specimen and test the direct specimen, we don't have to wait for it to grow. We can, in real time, what is here, right? And then you have lots of isolates you test at the same time. Okay, isolates B, C, and D all had the same thing. We have an outbreak, right, in this region, but A, Z, and F from these other hospitals didn't. Maybe it's floor 5. Floor 5's got bad hand hygiene. Just kidding, they don't. They're really good. That's the hemo floor. It better be good. Anyways, considered gold standard for validation of new phenotypic assays. So molecular epidemiology, an epidemic. We should know these things, right? COVID, come on. What's an epidemic? What's a pandemic? What's the difference? You shouldn't even have to look at these. What is the difference? Is what?
I have one.
Okay. Okay. So yes, an epidemic is more localized, right? It's the spread of something in a more localized fashion. A pandemic is where it goes. It doesn't have to be worldwide. It could be a large geographical area. Though, like Akron has an epidemic of measles. I have no idea. They don't. I don't think they do. But like, there's certain parts of like New York City that do have outbreaks of measles because of people who are not vaccinated. That is not a pandemic. That is an epidemic, right? They have certain pockets that they're seeing it actually being transmitted to other people. Epidemiology is the collection and analysis of environmental, microbiological, and clinical data, and then molecular epidemiology is a study of the causative genetic and environmental factors at the molecular level. So we're ascertaining the origin, distribution, and best strategies of prevention. Phenotypic analysis measures the biological characteristics of the organism, okay, such as growth requirements or genetic type. Genotypic procedures target genomic and plasmid DNA. Remember, what are the plasmids? They're a little blip. Little circles of DNA they can pick up and transfer to each other, and that's how they have, you know, that's how they mate. Transfer, they don't really mate, but like, you know, they take their little pill out of it. They can transfer it to somebody else. They're like, hey, bro, what's up? Take some of my genes. In microbiology, studies are performed to follow the spread of a pathogen within a hospital from actions of physicians and communities, and infectious disease specialists can determine genetic similarities and differences. So let's say there was a C. diff outbreak here, and let's say there was a C. diff outbreak at Summa. Is this a community outbreak? We can genotype both of those samples, those outbreaks, all of the samples, and see if they came from the same strain. Right? When they do E. coli testing and they determine there could be multiple outbreaks at the same time, which has happened, right? Let's say there's bad lettuce causing one outbreak, and there's bad beef causing another outbreak, right? You can sequence those, see if they came from the same source. If they're different, they can say, okay, these two species are different. Maybe they didn't come from the same source. Maybe this, and then they can like determine, right, where it might have come from back tracing. Okay, all of these people had this E. coli. What did they all eat? All of these people had this E. coli. What did they all eat, right? They can determine where it came from. Pulse field gel electrophoresis. So it's very similar. Digestion of genomic DNA, cultured organisms with restriction enzymes. You throw it through a gel. Changes are counted as gains or losses of bands or compared within the reference material, the outbreak material. So maybe this is the outbreak material. Okay. I don't know. Look. 3 matches. 4, 5, and 6 don't. Right? Different outbreak maybe. Okay. If 2, right? 1 and 7 are the ladders, right? If 2 is our outbreak, again, 3 is also outbreak. 4, 5, and 6 are not. They're different. They have different banding patterns. So, genetic differences. The rules of 3, okay? Indistinguishable is 0 genetic differences and 0 fragment differences. Test isolate is the same as the outbreak strain. Closely related. There might be 1 genetic difference, 2 to 3 fragment differences. Test isolate is closely related to the outbreak strain. Maybe it is related. Maybe after a few rounds of passing from one thing to another, it did slightly mutate, right? Like possibly related. Test isolate is possibly related to the outbreak strain. Again, maybe it's gone on for a little bit longer, and you might have more genetic differences. And then greater than 3 or greater than 6, test isolate is unrelated to the outbreak. So after you get so far removed, it's likely completely something different. So arbitrary prime PCR, randomly amplified amplification of polymorphic DNA. Okay, so you compare test strains to reference outbreak strains. The difference in band pattern equals differences in strains, and this method is more complex and difficult to interpret. Isn't this crazy? That's a lot of bands, right? So this is your ladder, by the way. Just that one. Who's related? Potentially. Oh my God, you counted already? Yeah. This one? This guy right here? Anybody else? I don't think so. So it could be more some that are closer related though, that aren't identical. So this one looks like it's identical, right? This one has 3 differences. Okay. So based on our rules, what are they? Possibly related. Okay. This one is, I don't know. We got lots of stuff going on. Some of them are more closely related than others. Okay, based on differences. Okay, so interspersed repetitive elements. Amplification from repetitive elements reveals differences in sizes of unique sequences. Okay, isolate A, isolate B. Okay, we have isolate A and isolate B. Which one has larger repeats? A. Move slower through the gel, and you can see, right? This is larger between here and here. Those are repeat regions. So if we're looking, that's same as like if remember Fragilites, we were looking at those TGG repeats, how many there were, right? Same kind of method, but in between other regions. We're talking about bacteria here. Same sample. You're right. And that could be so big or so small, right? Other genotypic typing methods. Plasmid analysis with restriction method, restriction mapping. So restriction, fragment length polymorphism analysis by Southern blot. You cut the DNA, you resolve the fragments by gels, you transfer the separated fragments to a membrane for probing. So you can identify specific sequences that you're looking for with specific probes. So you have amplified fragment length polymorphism. So you take your fragment length and you amplify it, right? You cut it with restriction enzymes. Your interspersed repetitive elements distinguish between bacterial isolates based on genomic locations of conserved sequence copies. Again, you might have one species always has 10 repeats, right? Another species might have like 40 repeats. You can identify differences based on the number of repeats. Ribotyping, these are ribosomes. Spot typing, 2 to 16 repeat units can be analyzed and compared to known isolates. And then multi-locus sequence typing, which is characterized isolates by using sequences of fragments of housekeeping genes. So housekeeping genes are what? We talked about this in us. Something that's there, it's regulatory, it's ubiquitous, right? So bacteria have the same thing. They have essential genes, something that's always there. But there could be slight variations in their genetic sequence. So we can also sequence things. Comparison of methods. So we have all our methods. And we have ease of use, ease of interpretation, reproducibility, discriminatory power, and typing capability, right? You have to look and weigh all of those methods to determine the best method for what you're looking for. This is not a one-size-fits-all box, right? If you are trying to be able to easily interpret something, you don't care the cost, right? The cost isn't a limiting factor. Maybe ease of use isn't a limiting factor because you got really expertise techs who know what they're doing, right? Maybe you want to go repetitive elements, okay? But you also, ease of use is important. You have to know the limits of the text and the technology that you have. If something is easy, but not maybe necessarily as sensitive, that you can get a result quickly, and you know it's going to be reproducible because it's not very hard of a technique, that might be the method for you to determine it. So also it depends on what level you're trying to discriminate the bacteria on or whatever that you're running. You might not care for whatever reason what subtype it is or if it came from the same species. But if you're looking for epidemics and pandemics and things like that and outbreaks, you definitely do. You want to look at that if you're trying to trace it to its source. So you have to look at all of those things. Yeah. These are two things that they use in micro. They use the biofire and they use the Cepheid. You'll learn about those. Micro is different than micro. Micro's 3 weeks. Viro's like a day. Virus, or was it 2 days? It used to be 2. Did they drop it down to 1?
Like 1 month. Yeah.
Because they don't do shell cultures anymore. It used to be 2 days back in the day, and we got to do shell cultures back in the day when I was here 2 years ago. I don't think they do it anymore. You're not going to get to do it unless they still have some hanging out for you guys just for like teaching purposes. I don't know. You'll only be in molecular for half a day though. It'll be grand. Um,